Hello everyone. Today we're going to have a brief introduction to parasitic protozoa. So far we've talked about the relationships between hosts and parasites. And so today we're going to look at our first class of parasites, which are protozoans. So parasitic diseases can occur pretty much anywhere. Particularly we see them a lot in rural or underdeveloped or crowded spaces. This graph right here is just a global representation of the global distribution of some of the parasitic diseases that we deal with globally. We mentioned malaria in our last class session, schistosomiasis, trypanosomiasis, leishmania. These are all some pretty common parasitic protozoa that we deal with across the globe. And you can see, again, this sort of a global distribution, all right? We see the malaria, schistosomiasis, trypanosomiasis. We see those across majority of the African country. We see a lot of trypanosomiasis in the Americas, the North America and South America. And over in Asia, parts of Asia, we see a lot of the leishmaniasis, and also in the northern parts of Africa. So we talked about some routes of transmission. When it comes to parasitic protozoa, we have some of the typical routes of transmission, the fecal-oral route. So in some way or another, an individual is ingesting, okay, by way of either contaminated food or water, okay. Some of those organisms will probably be the Entamoebas, Giardia, Toxoplasma, all right. We talked about the Giardia just a little bit in our previous lesson, where some of the side effects are basically it's a gastrointestinal infection as well as Entamoeba. Vector-borne routes of transmission, we talked about these vectors. For example, the mosquitoes, okay, as a vector. We talked about the vector as our vehicle, all right? These are the agents that are carrying or transmitting these particular parasites. So vector-borne routes of infection, sexual contact, okay, trichomoniasis. We mentioned that one just a little bit, okay? And then contact, okay? So these organisms are able to penetrate the skin directly, okay? So an example of that is schistosoma, all right? We'll look at quite a few diseases that we contend with due to infection with schistosoma species, all right? So these are all potential routes of infection for parasitic protozoa. So in this course, we're looking at the types of parasites that we deal with collectively, and we said there are three major groups of parasitic organisms, with the protozoa being the first, helminths, and then some arthropods. Okay, so let's look at some of these parasitic protozoa. All right, in terms of characteristics, I do want you to be familiar with some basic characterization of these organisms. Okay, these parasitic protozoa are going to be found in the kingdom Protista. Okay, Protista. So if I asked you to, provide some identifying characteristics, I certainly want you to be able to tell me about the domain, which would obviously be eukarya. These are eukaryotic organisms, okay? The kingdoms is going to be Protista, all right? So let's look at some basic characteristics of protozoans that you should certainly know at this level, okay? These are animal-like eukaryotic organisms, all right? We know what's characteristic of the eukaryotic organisms, basically the presence of a nucleus, okay? And some of the other membrane enclosed organelles, just depending on the organism. These are obviously going to be microscopic organisms. They're typically unicellular, okay? They lack a cell wall. All right, they are motile. They can move by way of the pseudopodia or flagella or the cilia. Okay, so some organisms, we can easily, identify them by the presence of these motility structures. Okay, this is our flagella right here. Okay, this organism has cilia. Cilia are basically these hair-like projections that stick out from the cell membrane of these organisms. And then we have this sort of amoeba-like structure right here that's moving by way of the pseudopodia, okay, those false feet that are giving this organism the ability to move. Okay, so we certainly want you to be able to, you know, tell us or understand that these are motile organisms. Okay, they primarily reproduce via asexual reproduction. Okay, so you should certainly be able to contrast, you know, two types of reproduction. There are a few that can reproduce sexually, but majority of these are going to have asexual reproduction. There's only one parent involved. Protozoans can be free-living or parasitic. We're particularly interested in those that are parasitic, and we're going to look at those in the acomplexa class. Okay, they're mostly going to be found in water and soil, and we're going to take a look at how the presence of these organisms in water and soil can become a potential problem. And particularly, again, when we think about those underdeveloped nations where, you know, there's a lot of poverty, there isn't great sanitation, you have a lot of individuals living on and off of the land that they live in, from, you know, fishing in creeks, washing their clothes in creeks and rivers and so forth. Those are potential routes of infection in those bodies of water and out in the soil. Okay. So these protozoans are can be symbiotic in our own animals, and some of them can cause human disease, which is why we care about them. We're going to take a look at some, again, that are pretty common and a potential human pathogen problem. Okay, so nearly all humans have been infected with protozoa at some point in their life. So this is anything that's abnormal. All right. Some of the species are commensals, and we talked about, again, those host-parasite relationships. Those that are commensal are present, but they're not causing too much problems in the host, right? But then there are some that are pathogenic, and those are the ones that we want to make sure that we, you know, fully understand and can characterize. All right. Infections with parasitic protozoa can range from being just, again, a mild asymptomatic infection to very life-threatening. Okay. Some common protozoan pathogens that you should be familiar with are Toxoplasma gondii, Cryptosporidium, and Giardia. All right, we're going to characterize these a little bit more in this lesson. All right, these are some common protozoan pathogens. Okay. All right, and so again, when we talk about parasitic protozoa, there are different ways that we can classify these organisms. Okay, one way we can classify these organisms or group them is by their mode of locomotion. Okay, protozoa can be identified based on their mode of locomotion. Okay, you have the amoeba, okay, that are going to be characteristic of having these pseudopodia. Okay, you have the ciliates. The ciliates are going to be those that have these cilia, which are the hair-like projections that project out. Okay, it aids in movement. And then you have the flagellates. The flagellates are going to be those that have this flagella present. Okay, we know that the flagella is this tail-like structure that protrudes from the end of the organism. Okay, so this is one way we can classify or group protozoa. Okay, we can also identify these organisms in their various lifestyles. Okay, those that enter the host enter humans through ingestion have two morphological forms. Okay, you should certainly be familiar with this life cycle and these morphological arrangements. Okay, they can exist in the trophozoite stage. Okay, this is going to be the feeding and reproductive stage that lives within the host. Okay, so they can exist in the host as a trophozoite, or we can find evidence based on the presence of their cyst. Okay, in this case, the cysts are going to be the infective form of the organism that survives in the environment. Okay, and so what happens is that these cysts undergo excistment when they're ingested, and then the developmental cycle continues, that these cysts develop into trophozoites, and then we kind of have this repeated cycle. Okay, so again, these parasitic protozoa can exist in two morphological stages, as a trophozoite and as a cyst. Okay, the trophozoite stage is going to be the feeding, the reproductive stage. This is the stage in the life cycle of this organism that they live and thrive inside of their host. Okay, during the trophozoite stage, these organisms can undergo encystment, okay, before leaving the host, and basically they're going to leave the host by way of being released in feces. Okay, so this organism is going to be encysted. It's basically going to develop into a cyst, and it's going to be released from the host in feces. Okay, and this is how it gets out into the environment. All right, the next life cycle, the next phase of the life cycle is that it will undergo excystment. Okay, so we have these cysts out in the environment. One way or another, it's made its way into our food or our water. We in turn ingest this infected water. And these cysts that we ingested in our infected water will now undergo excystment, okay, when it's ingested. All right, and these cysts will begin to develop into this trophozoite stage, and we'll have a repeated cycle. Okay, so we have two terms here that you should understand. Okay, that these organisms that enter the human through ingestion, okay, this life cycle, these morphological forms are specific to those organisms that enter the host through ingestion. Okay, eating or drinking. So you have the trophozoite stage and you have the cyst form. Okay, this is just some micrographs of what the trophozoite and cyst stage look like, and we can see some very prominent organelles here. For example, this nucleus. All right, these are eukaryotic organisms, so we know that the nucleus is a prominent feature. Okay, let's look at the amoeba. Okay, the amoeba is a very common protozoan. Okay, again, these are unicellular protozoa. They don't have any defined shape. Okay, this is another image of the of an amoeba. You see these pseudopodia sticking out. Okay, these pseudopodia are how amoeba move and acquire food. Okay, you can find these amoeba pretty much any water sources or in the soil. Okay, most of them are going to be commensal. However, a few of them can cause human disease. Okay, so the Entamoeba can be a human pathogen or is a human pathogen, as well as the Naegleria species. Okay, Naegleria and Entamoeba are examples of amoeba. Okay, protozoa that are unicellular. They're commensals. Usually they're not going to cause too much problems, but there are some that can be potentially harmful in the human. Entamoeba species. Okay, this one is going to be characterized by an infection with Entamoeba histolytica. This is our organism, Entamoeba histolytica. Okay, this type of parasitic protozoa is transmitted to humans by way of the fecal-oral route. Okay, and what does that mean? Fecal-oral route means that the individual ingested some contaminated food or water. Okay. This pathogen can be detected in feces of infected patients. Okay, so we're going to need a stool sample, and we're going to have to analyze that that stool sample for the presence of trophozoites or cysts. Okay, so actively motile trophozoites can be seen in a freshly passed stool. Okay, because remember, these are motile organisms. Okay, if the stool isn't fresh and it's had time to sort of form and get hard, it's usually going to be a lot harder to detect those trophozoites, but we can probably detect the presence of some cysts in the stools that are hard or formed. Okay, so this is one of the organisms in this class. of pathogens or parasites that you should be able to characterize and describe. Okay, the amoeba. All right. One example is the Entamoeba histolytica. Okay, it's transmitted by the fecal oral route of transmission. And so what does this pathogenesis look like? Okay, an infection with Entamoeba histolytica is that an individual is going to ingest the cyst. Okay, the cyst is going to pass to the stomach. All right, you know, you've got all kind of gastric acids and so forth in your stomach that's basically going to stimulate the exsystemate, which is going to be the release of these trophozoites into the intestines. The presence of the trophozoites are going to lead to some extensive damage in the intestines and could potentially lead to necrosis, okay, which is basically tissue destruction. Okay, so this tissue destruction is going to, you know, again, be due to the presence of cytotoxins that can be produced by these organisms. All right, so we're talking about major gastrointestinal discomfort. Okay, so again, these trophozoites or cysts can be released into the feces, and again, this is how, you know, it makes its way back into the environment, and this cycle of infection continues. Okay. So this slide right here is just a representation of the life cycle of Entamoeba histolytica, one of our prototypical amoebas. All right. So we have the two stages of life. Okay. We have the cyst form and the trophozoite form. Okay. So we said that the trophozoite form is basically going to be the form that persists in the host. Okay. Feeding is going to be multi-replications taking place. The cyst form is going to be the infective form. Okay, that's very important that you note the infective form for this organism is going to be the cyst. Okay, these cysts are going to be passed out in feces. They can be resistant to a lot of the antimicrobial agents that, you know, try to rid the presence. Okay, so the cyst is ingested. Okay, the trophozoite stage begins. Exsystement happens as the trophozoite makes its way through the stomach and into the intestines, and these cysts are released out in our stools and feces. Okay, and this life cycle continues. So do be able to describe what's happening here during this life cycle. Okay, so very simple life cycle with only 2 morphological stages you should be aware of. So some clinical symptoms of Entamoeba histolytica, amoebiasis is basically intestinal discomfort. You've got tissue damage in the large intestines, abdominal pain, cramping, colitis, diarrhea, dysentery. So we're talking about bloody stools here. In the carrier state, sometimes referred to as an asymptomatic state, an individual will not even know that they have the infection. Okay, the organism may be present and continuing to reproduce and produce these cysts. And these cysts can be passed out again in our feces without any true clinical symptoms. Okay, so there are a number of reasons why an individual can be asymptomatic. Okay, you should definitely note some of these. This is a very important concept. We talked about it a little bit when we talked about our host parasite interactions and how the host responds to an infection. Okay, here are some reasons that can potentially explain why an individual might be a carrier, but you know, asymptomatic, don't present any clinical symptoms. Could be due to a low virulence. Okay, the strain is not one that has potential for high or a lot of problems. Okay, so you have the ingestion of a strain that has low virulence. All right, low inoculum. Okay, so the amount that was ingested may not necessarily be enough to you know, elicit the immune response that results in symptoms. Okay, it may not be enough to, you know, cause all of the abdominal pain and cramping and dysentery. Okay, so the low inoculum. The inoculum is referring to what's being ingested, the organism that's being ingested. Okay, so it could be, you know, again, dose responsive. Okay, if you've only ingested a little bit, it's probably enough for your body to clear it without, you know, very many symptoms. Okay, and then lastly, an intact immune system. Okay, again, your body, you know, you're born with natural defenses. You have an immune system. Okay, that's the purpose of your immune system, is to detect these foreign pathogens again and rid the body of it. Okay, so in your notes, definitely be able to describe or identify explanations for why an individual may be asymptomatic even though they do have this infection. Okay, and this is true just in general. Like, why might an individual have this infection but they don't show any clinical symptoms? There could be a number of reasons. Particularly the strain that you're infected with has low virulence. Again, the inoculum size wasn't very great, wasn't large. Your body was pretty much able to clear it without too many problems. And then your immune system, you have an intact immune system. So long as you're not, immune compromised for a number of different reasons that a person can be immune compromised. Could be taking some medications like steroids. They can weaken your immune system. You might have some other underlying health conditions that renders you immune incompetent. Okay, pregnancy, other diseases, Crohn's diseases. There are a number of different illnesses and underlying medical conditions that can make you have a weakened immune system. So as long as you have an intact immune system, you potentially not show clinical symptoms of having the infection. And then the only way that we are able to even know that you have an infection is through, one of our diagnostic routes that we talked about. Okay, the stool samples, serology, and so forth. Okay. Another one, Naegleria fowlia. This is another example of an amoeba species that is a human pathogen. Okay, there are some additional readings on this species in your Blackboard course. Okay, so when you get a moment, do head over to Blackboard for some readings on Naegleria. Okay. Okay, so we just characterized the amoebas. Okay, we talked about, these parasitic protozoa can be classified into three different classes. We had the amoebas, the ciliates, and the flagellates. Let's look at the flagellates. Okay, structurally, these flagellates are going to basically have the presence of these flagella coming off. Okay, this image right here is Giardia. Okay, this is a very distinct morphological arrangement that if you see it, you should be able to identify these as Giardia species. All right, so these organisms are some characteristics are that they're small, they're single-celled, they have a leaf-like appearance. Okay, so this Giardia here kind of looks like a leaf. You got that stem in the middle. Okay, these organisms are going to contain at least one flagellum. Okay, we know that this flagellum is important in movement or locomotion. Okay, the number and arrangement of flagellum helps to identify the species. Okay, so you can see here some very distinct, projections of flagella here, flagella on this organism. There are 5, 2, 4, there are 6, pardon me. Okay, so the number and arrangement, are characteristic of a species. We can use that to help identify a species. Okay, if we were trying to analyze a stool sample and we're looking under the microscope, we can look for certain distinguishing features. Okay, one of those being the number of flagella that are present. Okay, these species can be found in water sources like streams, lakes, out at mountain resorts. Okay, some common examples of flagellates are going to be our Trypanosoma, Leishmania, and Giardia. Okay, make a note of these. Okay, flagellates. These are basically going to have these leaf-like appearance. They're going to have at least one flagellum. Okay, and some examples include Trypanosoma, Leishmania, and Giardia. Okay, make sure you jot those down in your notes. Let's look at one of our characteristic Giardia species, Giardia lamblia. Write this one down, Giardia lamblia. Okay, the cyst and trophozoite phases can be detected in fecal specimen from infected patients. Okay, this one again is going to be transmitted through fecal oral route, which means that an individual acquires this infection by consumption of inadequately treated contaminated water, ingested of contaminated foods. So these undercooked foods. That's why it's important to, cook your foods to the proper temperature, okay, to potentially kill any possible pathogens that might be present, okay? And it can also be spread through person-to-person contact. So down here is another image of what these Giardia species look like underneath the microscope. Okay, again, we can look for, again, those defining features here, like this just morphological structure in general, kind of looks like a leaf off of a tree. And then we have our projecting flagella here, okay? And so Giardia lamblia, this is definitely one of your characteristic protozoa that you should know, okay? Pathogenesis with Giardia lamblia, okay? What does this infection look like? You're ingesting the cyst. You're ingesting these cysts basically in your contaminated foods or water, okay? That water wasn't properly treated or your food wasn't properly cooked and that organism survived, okay? Or the cyst, you ingested these cysts, okay? Same process, you got the cysts that are gonna be ingested. Gastric acids are basically gonna stimulate that process of excistation, okay? Excistation is gonna release these trophozoites into the intestines. They're going to go through their reproductive stages and multiply, and it's going to result in local tissue damage. Okay, so you should definitely be able to characterize an infection with Giardia lamblia. Okay, what does this infection look like? What's happening? How did the individual become infected? Okay. Another graphic here just showing you morphology of this trophozoite phase. Okay, this is the phase again that they're persisting in their host. And then we have the cyst phase. This is the phase at which they're released in the stools, okay, in feces. Now this is just another representation of what's happening during this life cycle with an infection of Giardia lamblia, okay? So the first step here is that food and water that's contaminated with these cysts are consumed. You consumed some contaminated foods or water, okay? So you ingested the cyst. These cysts must go through the process of excistation, okay? Make sure you can compare and contrast what's happening with excistation and encystation, okay? Multiplication is going on right here. These organisms are reproducing, okay, making more. The trophozoites are going to colonize the small intestines, okay. Early stages of encystation takes place where the new cysts are going to be formed, okay. So the cysts are released here, replication takes place, and encystation begins, okay. New cysts are being formed, and these new cysts are going to be released in feces, okay? This is a very great representation of what's happening, and you should be familiar with this life cycle here. So clinical symptoms of a Giardia infection. The clinical diagnosis is giardiasis, symptoms of giardiasis. These are in individuals who are symptomatic. You're going to have a sudden onset. It means it's going to happen quickly, pretty much not long after you ingest those contaminated foods or waters, okay? You're going to have a foul-smelling, watery diarrhea. You're going to have abdominal cramps. You're going to experience some flatulence, okay? You're going to be gassy, cramps, intestinal discomfort, okay? Most or about half of the people who are infected are going to be asymptomatic, Okay, you're not going to have a whole lot of clinical symptoms. Okay, again, and we just described reasons for why an individual may be asymptomatic. Okay, be clear about that. All right, but giardiasis, an infection with the Giardia lamblia, is basically going to present itself as intestinal discomfort, diarrhea, cramps, flatulence. Okay, and it typically happens quickly. All right, our last class of protozoa are ciliates. Okay, ciliates are going to be characteristic of these hair-like projections coming off the sides of this organism. Okay, these hair-like projections are called cilia. Cilia, they aid in motility, locomotion. Okay, this is another way these organisms can move. All right, the cilia are typically present in very large numbers. Okay, these organisms are going to reproduce asexually, asexual reproduction. And again, they can be found pretty much anywhere there is water. Okay, so we're talking about lakes, ponds, oceans, rivers, and then again, obviously in our soil. Okay, ciliates. This is our last class of parasitic protozoa. There aren't a whole lot of them that are human pathogens, but there's one that we need to be familiar with, and that's Balantidium coli, B. coli, okay? Not to be confused with E. coli, Escherichia coli, okay? B. coli, Balantidium coli is the only ciliate that has been identified as a human pathogen, okay? You should be familiar with this organism, okay? Infection with this species is associated with contamination of water supplies with pig feces. Okay, so somehow or another, pigs have ingested and become contaminated with it, and releases their cyst out in their stool when they have a bowel movement. Okay, so water supplies that have come in contact with this infected pig feces can be one of the roots of infection here. Okay, it can also be spread through person-to-person contact. Okay, so pathogenesis with Balantidium coli, B. coli, ingestion of the infected cyst. Okay, so this is a common. So hopefully we're seeing our pattern here for how these parasitic protozoa becomes problems. Okay, basically through ingestion, right? Ingestion takes place, okay, ingestion of these infected cysts. Extusation takes place. Those cysts are basically going to turn into trophozoites, and the trophozoites are going to invade the mucosal lining of the large intestine, okay. Reproduction takes place, incestation takes place, new cysts are being formed, okay, as these organisms, these trophozoites are making their way through the intestines. Okay, new cysts are going to be formed, and they're going to be passed with feces. Okay, so we should definitely have identified our pattern now. All right, we ingest the contaminated cyst. These cysts are going to go through extusation. Okay, reproduction takes place. More development, reproduction. Trophozoites form. New cysts are going to be made. And these newly encysted cysts are going to be released or passed through feces. Okay, there's another schematic of what's happening with our B. coli infection. The cyst is the infectious stage, right? So we set that, and it is acquired by the host through ingestion of contaminated foods or water. Okay, this is a nice schematic that I found from the CDC, okay, from a B. coli infection. The cyst is the infected form. You ingest the infected cyst by consuming contaminated foods or water, okay? The trophozoites are going to invade the intestines or colon, large intestines, and new cysts are going to be formed, all right? And then those new cysts are going to be released in feces. All right, oops. All right, so clinical symptoms of B. coli infections: balantidiasis, okay, abdominal pain and tenderness, nausea, okay, anorexia, watery stools with blood and pus, so dysentery, ulcerations, of the intestinal mucosa, so possible ulcers are forming in the intestines, okay? This can be a very serious infection, all right? Asymptomatic individuals, most cases are going to be asymptomatic. But again, if an individual who, for example, has a weakened immune system, they are going to experience some pretty severe symptoms, okay? Like the abdominal pain and tenderness, the nausea, you know, the watery stool, watery bloody stools, ulcers forming, okay? It's pretty painful, okay, if you are presenting symptoms here. Okay, so to recap, we talked about, we introduced our characteristics of parasitic protozoa. All right, you should certainly be able to identify some basic characteristics in terms of identification or taxonomy. All right, eukaryotic, unicellular, you know, so forth and so on. And then I gave you some ways that we can identify them based on their modes of locomotion, okay? Do they have flagella? Do they have cilia? Do they have pseudopodia? Okay, so we have the amoebas that have pseudopodia. We have the ciliates that have the cilia, the hair-like projections. And then we had our flatulates. Those are going to be the ones that have the flagella, one or more flagella present, okay? And so that's another way that we collectively can identify protozoa is based on their mode of locomotion, okay? And we gave you some representative organisms in each group, okay? We gave you some representative flagellates. We gave you a representative amoeba, the Entamoeba histolytica. We gave you some representative cilia, and which the B. coli, and some a representative flagellates, the Giardia lamblia. Okay. Talked about diagnosis a little bit previously. How do we diagnose protozoan infections? Okay, certainly microscopic examination of feces. Okay, so we're looking for either the evidence of trophozoite or those cysts. Okay, some can appear in showers. Okay, so we might not be able to just take one stool sample. We might have to take several stool samples over time. Okay, because the presence of this organism, for example, Giardia, appears in showers. Okay, so you might have a bowel movement at 2 o'clock that has zero evidence of this infection. Okay, but when you have a bowel movement and another couple hours, say at 4 o'clock p.m., we may be able to identify the presence of this organism. Okay, and so for this reason, because some organisms appear in showers, we may have to collect several stool samples over time in order to make a proper diagnosis. Okay, if you go and see your primary care provider and you're having this unexplained intestinal discomfort and so forth, and we collect a stool sample and don't find anything, we can't quite rule out these parasitic protozoans right away. Okay, because some of them just may not have presented right now at that time. Okay, you do have an extension exercise. I'm going to stop right here.